Hello, and welcome back to this scientific ethics lecture where we're actually going to start looking at recombinant DNA, start looking at this idea that this DNA is going to be important, and if we can go through and make changes to the DNA, we can change what's going on within the organism, or at least within the organism's cells. And so part one of our discussion of recombinant DNA, we're going to take a look at genetic engineering, or basically how is it that we can go through and start manipulating or changing the DNA. So kind of as a quick reminder, I'll remind you that the DNA is going to be very important because the DNA holds that information that is key to the understanding of what is going on within that cell, within that organism. And then that DNA is going to be passed from the offspring. And so the genes, that specific information that's carried within the DNA molecule, is going to be passed from the parent to the offspring. And it's going to be these genes, what's coded within the DNA, that information within the DNA, that's going to determine the characteristics of the child. And that was the whole basis of heredity. And it's the whole basis of the reason why you look a little bit like your parents, probably a little bit like your siblings, but you're probably looking more like your relatives than the general population in comparison. And the reason for that is that you're going to share more genes and the characteristics of those genes with people that are closely related to you than people that are further away. And so again, similar to those DNA kind of similarities we talked about when we were trying to do DNA fingerprinting, identifying individuals based on their DNA, things like that, the same type of thing is going to be occurring when we take a look at passing these characteristics from parent down to the offspring. Now, if we take a look at this, over thousands of years, over even millions of years, we want to extend it that far, living organisms have changed dramatically over generations. And one of the main reasons for that is the process of evolution, but humans have impacted on that. Humans have gone through and what we've described it as domestication. We've taken wild things and domesticated them, made them kind of less wild and more towards what we're interested in looking at. And what we're going to be doing is a process of artificial selection. In artificial selection, we're going to go through and do selective breeding for desired traits. Again, based on that idea that offspring are going to look more like their parents than the general population. And so a good example of this is if we take a look at this nice full corn cob here. We can see kind of the modern day corn. We got a cob here kind of underneath, and then we've got rows and rows of these filled, corn kernels. And they're filled with nutrients present within them, and they're a great food source. But if we take a look at our modern corn cob and take a look at it kind of in a historical perspective, modern day corn was transformed from a wild plant called teosinte. Teosinte looks like this over here. And so if you can take a look at this, you can see, 1, 2, 3, 4, 5, 6, maybe a dozen kernels of corn on this. And it's kind of hard kind of imagining, putting butter and salt on this and gnawing on that like you're going to gnaw on an ear of corn over here. But what happened in that process of domestication, what we got over here, is we're going to go through, and over the history of time, we're going to take this teosinte plant and we're going to breed it year after year after year after year. But we're not just going to breed it wildly. What we're going to do is we're going to take a look at what plants have the largest cobs and the most kernels, and we're going to grow those plants. And we're going to allow those plants to reproduce with one another. And so that next generation is going to be more like those that have lots of corn kernels and a bigger cob. And we're going to take those and we're going to breed those together. And it's gradually going to get bigger and bigger and bigger and more kernels associated with it. And so we've changed both the genetics and the outward characteristics of corn from the original teosinte plant by selective breeding, by choosing the parents so that their offspring are going to be more like what we want it to be, looking for those desired traits. So we get a good full ear of corn over here. And so that's one example of it. We can take a look at other examples as well. The wild mustard plant, you can take a look at it over here. It's something that, take a look at. If it's growing in your garden, you'd probably, kind of dig it up and throw it away, but pretty much ignore it. But that plant, that wild mustard plant here, through that process of artificial selection, selective breeding, has developed into all of these different types of vegetables that we see that are available to us. And so a wild mustard that was grown for generation upon generation upon generation, suppressing flower development, would develop into broccoli. Doing the wild mustard, growing it generation after generation after generation with sterility of flowers is going to give us the cauliflower. If you suppress the height of the plant, you're going to end up with a cabbage plant. Or if you want larger leaves, you can end up with kale. And then, kohlrabi, you're doing some other things as well. But basically, all of these vegetables that we'd recognize and we would think about as being very, very different flavor, very different uses in our diet, take a look at it, very, very different in appearance, very, very different kind of overall. They're all derived from a wild mustard plant over thousands and thousands of years of selective breeding, small changes over a long period of time. And so again, we're looking at changes in those outward characteristics based on the changes of the DNA. And again, we're changing that DNA by choosing the parents that are going to be breeding for that next generation with the idea that the offspring are going to be closer to their parents, to that wild population. And so if we reinforce that time after time after time, generation after generation after generation, we can get very small changes resulting in very big changes. And what we'd recognize as being different types of vegetables in this case. And we can look at it in animals as well, again, through artificial selection. Our modern day dog is descended from wild wolves. And so through that process of domestication, they were able to take wild wolves and tame them. But selectively breeding them, they were able to generate a wide variety of different breeds of dog, all the same species now, so they can still interbreed if we take a look at it, so they can still mate and it's kind of challenging. You got a big dog, a little dog, they can still breed and have offspring. But in general, what we're looking at is very, very different characteristics, again, because of those changes that we made in their DNA over a period of time. And the size and the characteristics of the animals is based on different goals. Some of them are going to be hunters, so they're going to be good at, seeing a prey or smelling a prey and chasing it down. So they're going to be very, very good. You can look at, little terriers, which are small dogs, kind of similar to this, that aren't, hunting, big animals, but they go down and they kill rats. You can find other dogs that, you use on a farm to herd sheep. So they're not going to be hunting, but they're going to be very good at, tracking sheep and keeping them all in a cluster. And then finally, you've got companion dogs. In many cases, dogs that are bred for a very good temperament, oftentimes a little bit smaller than some of the hunters that you've got out there. But again, very, very different. You look at all the different breeds of dogs, it's hard to believe that they came from the same descendant. They came from the same wild wolves, because through that process of artificial selection, through that process of selective breeding, the offspring, like their parents, and we're breeding the parents based on some characteristic that we're interested in and we want to reinforce, we're able to very, very dramatically change them. Still the same species, but they look very, very different. Another example of that is a mule. And so if we take a look at this, a mule is a hybrid offspring between a male donkey and a female horse. And so if we take a look at this, a horse has two sets of 32 chromosomes, a donkey has two sets of 31. And so if you breed a male donkey with a female horse, what you end up with is this mismatched set of chromosomes. You've got 32 chromosomes from the horse, 31 chromosomes from the donkey. It's mismatched, so these organisms are going to be sterile. So you can ask why we do that. You take a look at it. This mule over here is going to be faster and more intelligent than a donkey, but it's going to be more patient and long-lived than a horse. So you're getting better characteristics than a true bred horse or a true bred donkey by crossbreeding, producing this hybrid. And again, looking at the characteristics that we're interested in, breeding it, and seeing the characteristics that we want. So all of the examples we've talked about so far are going through and using a natural process, choosing the parents to mate, to have offspring, in the hopes that those offspring are going to be more like their parents, are going to have characteristics or traits that we're interested in. What we're looking at is doing this, and instead of over thousands of years and generation upon generation upon generation, we're looking at doing it much faster in a laboratory setting. And so what we're going to be doing is looking at this concept of recombinant DNA or genetic engineering. So recombinant DNA is basically recombining. So recombining DNA from two or more sources into the creation of a new combination of a DNA molecule. And so a good example of this in this slide here is that we've got a vector, and again, don't worry about some of these scientific terms that I'm giving you. A vector is basically like a carrying case for our DNA molecule. It has some characteristics that allow it to work and copy itself within a cell, but it's also possible to take fragments of DNA and incorporate them into our vector. Now, if we do that, we combine our fragment of DNA with our vector DNA, our carrier DNA. We've now recombined it from two sources into this recombinant DNA molecule over here. So we're creating, in a laboratory setting, new combinations of DNA that under normal circumstances are not normally going to be found in nature, but we're going through and we're going to be engineering it. And so this process of genetic engineering is this idea that we're going to have the equivalent of kind of molecular tools. We're going to have molecular pliers like here, little molecular scissors, little molecular, like a soldering gun or a glue gun or something like that, is we're going to go through and we're going to be able to produce recombinant DNA. And again, recombinant DNA, just recombining sources of DNA, two sources into a single one. So we're going to have genetic engineering being that technology that allows us to isolate and manipulate the DNA and use that in some way, change it, and hopefully by changing that DNA, change the characteristics of whatever it is they're working with. And this has applications that we'll talk about later on today in medicine, agriculture, industry, kind of a whole range of things, as well as just scientific research. Now, if we think back, remember those restriction enzymes that we talked about when we were doing DNA fingerprinting? We said that they're going to go through and they're going to cut the DNA molecule into fragments. We're going to use those same restriction enzymes to go through and cut the molecule in specific locations. And the important thing is that they're going to cut it at a specific site, but in most cases, they're going to result in kind of the staggered cut here so that we have this kind of almost like jigsaw puzzle piece that we can work with. And when we do that, we can take a look at it. So we've got, our DNA molecule here. We can go through, and the EcoR1 is kind of this enzyme. That's going to be an example of one of our restriction enzymes. We can go through and cut it. It cut it kind of unevenly. So we've got this staggered kind of what's referred to as sticky end. So, sometimes scientific terms make sense. We got a sticky end here. But if we take a look at this sticky end or jigsaw puzzle piece is going to match up with this sticky end or jigsaw puzzle piece. And so whether we open up the same molecule and close it together, or we open it up and mix it with a jigsaw puzzle piece from another puzzle, we can put it together. They have the same shape, which is going to allow them to do this. And so we've got one source of DNA here, another source of DNA over here. We cut them with the same restriction enzyme. We kind of mix them together. And now we got this recombined or recombinant DNA molecule over here. And so again, using these enzymes allow us to make jigsaw puzzle cuts into it and then put these pieces together and come up with this recombined or recombinant DNA molecule. Now the vector I said was going to be like a carrying structure for the DNA. So it's going to be very important because it's going to have some of those restriction enzyme sites so that if we go through and cut it here, it's going to open it up. If we take our DNA fragment here, this little red piece, we cut it with the same restriction enzyme, we mix these two things together, and now all of a sudden, we've got our vector for carrying our DNA molecule with our new piece of DNA, our recombinant DNA, our red source of DNA, our kind of white source of DNA. We've recombined that into this structure. Now, the nice thing about the vectors is they can replicate in the host cell, which is a complicated scientific way of saying that if we put them into like a bacterial cell or into an animal cell, they can continue to copy on their own. So we don't have to go back into the laboratory and keep doing it over and over again. And so because of that, we put it in and it basically kind of one and done. We put it in, it's going to be able to do what it needs to do and keep going as long as that cell's alive. They often have a selection marker so we can figure out, okay, where is this? Make sure we isolate it and inject it into the host cell that we're looking at. And so the selector marker isn't important. It just means that we have a way to identify our recombinant DNA molecule when we're working with it. And so if we take a look at this, the most common plasmids, the most common vectors that at least were used kind of historically and traditionally are bacterial plasmids. These are little bacterial mini chromosomes. They're small circular DNA molecule that were extra within the bacterial cell. And because they're extra, they're not required, but we can go through and we can manipulate them. And so we can go in and insert in our DNA that we're interested in and then put it back into a bacterial cell, and all of the cells that that bacterial cell produces, when it divides and produces more and more cells, all of them are going to be carrying our recombined DNA molecule. And so we're going to be able to use that to make lots and lots of copies of the DNA that we're going to be interested in studying or using in some way. And so again, what we're going to be doing to make our recombinant molecule is going through, taking, in this case, we got the yellow vector here, we've got our blue piece of DNA there. We're going to cut it with a restriction enzyme. It's not showing up as well as I'd hoped, but you can take a look. You've got the sticky end here. We got the sticky ends over there, a little bit more clear. We mix these two things together. We end up with this recombined DNA molecule with the yellow DNA and the blue DNA coming from two different sources. So we mix them together. We use some enzymes to kind of seal it all up. And now we've got our recombined or recombinant DNA molecule. So if we take a look at this, only some of these recombined molecules are going to have what we're looking in. And so we're going to look at the markers. So if we take a look at this, this is a plastic dish. It's got some nutrient food source referred to as agar on it, and we're growing bacteria. And so if you take a look at each one of these little clustered kind of clumps of things, these are all going to be bacteria that are coming from a common progenitor, common ancestor, if you want to think about it that way. And so we can take a look at this, and what we can see is that we got some that are white and some that are blue. And so we've got, again, some of those markers that we can look at, and we can go through and say, okay, we're interested in the blue one. And so we're going to go through and we're going to isolate the blue colony of bacteria, and we're going to take it, we're going to transfer it, and we're going to grow it someplace else. Or we may be interested in the white one. So we're going to isolate the white one, grow it up in a separate dish. But we have a way in which we can find out which are the bacteria that are carrying our recombined or recombinant DNA molecule. So again, I'm emphasizing this because I think it's important to be able to understand how it is that we're using this technology and applying it to make changes to the DNA. So a DNA clone, if you think about a clone is going to be a genetic something that's genetically identical to something else. So a DNA clone is going to be a sequence of DNA that is copied by the host cell. And so we've got a bacterial cell over here. It's got its red normal chromosome, circular chromosome being present, and it's got some of these little blue plasmids. These are like the little vectors that we're going. Now the bacterial cell, bacteria reproduce asexually. They don't need a partner to reproduce. They basically are going to divide in half, and we're going to have 2 offspring, and those are going to divide in half, and we're going to have 4, and that's going to divide in half, get 8, and so on and so on and so on. But these plasmids are capable of copying themselves over and over again within the cell. And so what's going to happen is that when that cell divides, each one of those kind of daughter cells, if we want to think about it that way, is going to have the bacterial chromosome, the bacterial DNA, the red one here, but also going to have copies of those plasmids as well. And therefore, all we have to do is grow the bacteria. And growing bacteria is relatively easy compared to going through and isolating DNA and growing DNA, copying it, all that stuff. Is that we just copy or grow the bacteria, and the bacteria copy the DNA for us. And so what that means then is that we can end up with lots and lots of these bacterial cells. All of these bacterial cells are going to be carrying our vector and carrying our piece of recombined DNA. And we can use that then to get this bacteria to change its characteristics, to change what it's doing. And in many cases, we can get this bacterial cell to read what's within this DNA molecule and use that information to produce it. So we can look at an example of that in a few minutes. So again, we've got this mechanism where it's very, very easy to go through and get a bacteria, get an animal, or not an animal, get a living organism that's capable of dividing it and making the copies themselves. We'll talk about animals in the next set of lectures. Now, more recently, I'm talking within the last 5 to 10 years, there's a great and exciting new technology called Crispr, which allows us to go through and instead of using those restriction enzymes that we talked about before, they're going to be going through and again, I apologize for some of the science and the terms. I want to put it out there, but don't worry about it. CRISPR stands for Clustered Regulate Interspersed Short Palindromic Repeats. That's a mouthful. Just think about it as CRISPR is allowing us to go through and locate specific regions of the DNA molecule. It also has a Cas9 enzyme, which is going to be an enzyme that goes through and cuts the DNA molecule, opens it, and allows it to do something. Now, this CRISPR technique is basically allowing us to go through using some science technology. And instead of just looking at kind of where we have a restriction enzyme site, these locations where we might be able to use the existing enzymes and go through, this technique allows us to go through and basically at any point in the DNA molecule, go through and edit the DNA, go through and make changes to the DNA. And this is a technique, a process developed by Emmanuel Charpentier, Jennifer Doudna, and Feng Zhang. I'm only emphasizing this because think about these names. This is Nobel Prize level work. I would not be surprised if within the next 3 to 4 years, maybe a little bit longer, these 3 people are over in Sweden, assuming we get to travel again at some point in the future, accepting a Nobel Prize. This is how significant this advance is. So what we're looking at, again, we've got this DNA molecule here that we're looking at. And basically the way that CRISPR works is that we're going to have this enzyme, this Cas9 enzyme, which is basically like the enzymes that we talked about in proteins in the last lecture. This is like the worker. This is going to be doing something within the cell. We can put it together with a guide piece of RNA, a single-stranded nucleotide. And based on that sequence of nucleotides here, it's going to match up very specifically to a location within our DNA molecule. So it's basically going to take our Cas9 enzyme, our protein here, and attach it to our DNA molecule at a specific site, as opposed to just trying to find where one of those restriction enzyme sites works. So by changing this guide RNA here, we can have this attached to any point within our DNA molecule. So once we do that, the Cas9 protein is going to be lined up based on that guide RNA up here, is going to cut the molecule, and in cutting the molecule, it's going to open it up, and we can insert in our desired piece, and what's described here is programmed DNA, but this is our no source of DNA. So again, we got one source of DNA here, another source of DNA here. We've got this recombined or recombinant DNA molecule produced by mixing together 2 sources of DNA. And so we've got this recombinant DNA molecule that's going to be important. So if we take a look at this, and again, this is why this is so monumental. This is so important. CRISPR applications are going to allow us to go through and do all kinds of different things to work with the DNA, to make changes to the DNA, to potentially make genetically modified organisms, to treat disease, to screen drugs, gene therapy, disrupt pathogens, target RNA, all of these different things. It's incredible, incredible advance. And like I said, I expect in very short time, those 3 are going to be nominated or receive a Nobel Prize. Okay, so that finishes up our kind of introduction to how we do recombinant DNA. Again, broad overview, but hopefully enough to get you a good flavor for how these things are going to be working. just see a quick typo, correct. So that's how the DNA works, recombinant DNA works. Come back for part 2 of this lecture series. We're going to take a look at specific examples of genetic engineering. As always, if you have any questions, feel free to email me. Thank you and have a great day.